Hi, everybody. This is a particularly exciting EMPH seminar. Of course, it's Darwin Day week. So all things Darwin, all things evolutionary are on the go. And we have the amazing Athena Aktipis here today. Before I get into that, though, I just want to do a little bit of housekeeping. We do have question time afterwards, live questions. So please type any questions you have into the chat box on YouTube. If you do not have a YouTube account or you want to remain anonymous, you can ask a question through the Google Doc, which is also linked there. Great. Okay, so Athena is one of those people that doesn't really need an introduction, as they say. Her work is really broad. She's interested in cooperation more generally among people, but of course, thinking about cancer, this is really pertinent. And today she's going to talk about this amazing book, which really is an accessible way to understand, the evolutionary process that is cancer. Athena is also the director of various projects and founder of the Zombified Media and Zombified podcast, which I really love. So I'm not going to go on anymore. I will hand over to Athena. Thank you.
Paula, thank you so much for the invitation to speak, and thank you to all of you for being here. It is a real pleasure, and I'm just delighted to have the chance to talk to you all, and I'll look forward very much to the comments at the end. So I'm going to try to keep my talk to about 45 minutes so that we have plenty of time for discussion. So as Paula mentioned, I wrote this book called The Cheating Cell, and really it's about applying cooperation theory and evolutionary biology to understanding what cancer is, why it exists, and why it really is going to be with us for the long term. That cancer really is not just a modern phenomenon. It's something that's really built in to multicellular life itself. And I'm at Arizona State University. I do a lot of things here. I direct the Interdisciplinary Cooperation Initiative. I'm also a professor in the psychology department. I co-direct a lot of research projects, and I work on questions in evolution and medicine as well, applying the sort of cooperation and conflict framework to trying to understand, really what the processes are that lead to the evolution of vulnerability to disease, and also the evolution of mechanisms to help protect us from disease. And for about 15 years or so now, cancer has been really the focus in my work on evolution and medicine. And this book kind of represents the culmination of a lot of time and energy and work that I spent really just trying to get these frameworks from evolutionary biology, from ecology, from the sort of general science of understanding cooperation, and use those lenses to better understand cancer. So I want to begin, though, with this lovely, beautiful, mutated cactus. This is a crested cactus from the garden that we built here on the ASU campus. It's called the Endless Forms Most Beautiful Garden. And it is a garden that has both normal plants in it and these fasciated cacti. Fasciation is the sort of scientific name for this process when you get mutations in the growth tips of plants that lead to these sort of fans and other interesting and often really beautiful formations. And I'm putting this up here because it tells us two really interesting things. One is cancer and cancer-like phenomena are not unique to humans. We see them in other forms of life as well. And in fact, we see them across all branches of multicellular life. And the other thing about these crested cacti is that they show us that it's possible to live with cancer or cancer-like growths. So we created this garden to make it also more accessible to general audiences, to broad audiences, to engage about what cancer is and the fact that cancer is something that that we experience, that organisms experience across life. We worked with groundskeepers and gardeners, a master gardener, to design and put this garden in. And during that process, we also found that cancer touched many people's lives. Many of the people who were involved in the process of putting this project together had been touched by cancer, and it was really powerful for all of us to, realize how it does make a really big difference when you take the time to connect with general audiences and with people who are working in other domains about issues around cancer. And this deeper understanding of what cancer is something that I think can be very comforting for people who are dealing with cancer in one way or another. His work was covered in Phoenix magazine, and it's been a really wonderful thing to have here on campus, to be able to have events and bring members of the public and patients and patient advocates. So now bringing that back to sort of the scientific questions here, yes, we're not alone in our struggle with cancer. Cancer is something that has been around since the origins of multicellularity. And it's a representation, really, of a very fundamental tension. So in my work, I look at cooperation across systems. And in particular, I look at issues that happen when you scale cooperation up, when you go from having a few individuals who are cooperating and coordinating to having large groups, whether we're talking about a few cells and the evolution of multicellular bodies with, trillions of cells, or if we're talking about humans living in small groups, small scale societies, and then needing to regulate behavior to scale up to large scale global societies. Many of the same opportunities present themselves as you increase group size, right? So group size increases because there are a lot of benefits that can come from that. You can produce public goods in more efficient ways, sharing the benefits of them. You can achieve what are called economies of scale, where basically you're able to do things more efficiently with a larger group. You can manage risk more effectively with larger groups, and you can also divide labor. So, make it possible for some individuals to specialize in some tasks and others in other tasks. But there's a bit of a catch, which is as the group size increases, it becomes easier and easier for individuals who might exploit those systems to take advantage. It becomes, as groups are larger, it requires more effort and energy to be monitoring because you have more individuals who are there. And as groups get larger, they also often become more complex. So it can potentially be harder to tell whether cheating is occurring because there's so many complex behaviors that might be going on in these large scale systems. So in order to capture these benefits that can come from increased group size and cooperation, there have to be some mechanisms that solve these problems of detecting and suppressing cheaters. And so that's broadly a lot of what my work has sort of focused on. And in particular, when we apply this lens to cancer, it helps us understand how cancer is a fundamental vulnerability of multicellular bodies because we are made of all of these cells that have to basically do these processes of detecting and suppressing cheating in order for us to be viable. A little bit more about kind of how I got to this point of studying cooperation and studying cancer. for my PhD, I was at the University of Pennsylvania in the psychology department, where I actually did my dissertation using computational modeling, using agent-based modeling of the evolution of cooperation. This is a set of screenshots from the group-wise walk-away model, which was one of the topics of my dissertation. And with models like this, it's possible to put in cooperators, put in cheaters, and then see, what simple rules allow cooperation to evolve and to be viable. After that, I went to the University of Arizona, which, for those of you who are not from Arizona, the University of Arizona is in Tucson. And I'm here at Arizona State University, which is in the Phoenix area in Tempe. I'm sort of rival school. So I did my my postdoc at the University of Arizona in ecology and evolutionary biology. And I might not be a loyal ASUian for having done that, but I do love Arizona, and not just because of the amazing crested cacti here, but it really is a lovely place to investigate questions around the evolution of cooperation and multicellularity. While at University of Arizona, I had the pleasure of working with John Pepper and also with Rick Michaud, who's worked on the evolution of multicellularity. And he and I never published together, but I went to a lot of his lab meetings and talked with a lot of the graduate students, and we had a lot of really interesting conversations about the evolution of multicellularity and cancer. We're quite putting a lot of these ideas together. After that, I went to UCSF, where I co-founded the Center for Evolution and Cancer. I was so compelled by these questions about, how does cancer sort of represent this breakdown of multicellularity, that I shifted a lot of my research program while I was a postdoc to those questions. And then I went to the Wissenschaftskolleg in Berlin, the Institute for Advanced Study there, as a fellow, as part of this Cancer Evolution Working Group. And that's where a lot of this work was done, with many of my colleagues looking at cancer across the tree of life. And it jump-started, probably more than a dozen projects for all, across I think there were 5 of us, officially 6 of us, maybe, in the working group. And now I'm here at Arizona State University, where, among other things, I'm part of the the Arizona Cancer Evolution Center, where we look at many different aspects of cancer evolution, including somatic evolution, so how cancer is evolving inside the body, comparative oncology, looking across a species at cancer susceptibility. And then we also do a lot of outreach and education. So things like the book that I wrote, but also we have graduate students and some professors who go into schools, work with students. I write lots of pieces for general audiences, and we create art exhibits as well to engage people. And in a way, our garden is a sort of combination of a sort of general outreach project, and it's almost like a museum that's always open. So I wrote the cheating cell because I wanted to not just give people who were interested in evolutionary biology insights into cancer, or vice versa, but I wanted to also write a book that would make the science of cancer accessible to broad audiences. So I tried to really, do kind of a difficult thing, which is to take the complex science underlying evolution in cancer and write about it in terms that would be accessible to all of those audiences, and not sacrifice the complexity and the sort of intrigue, I think, that comes from a lot of these sort of deeper issues, like exploring, what kind of evolutionary trade-offs are there with cancer susceptibility? Why hasn't evolution eliminated cancer? And it was really a great opportunity to kind of think about the broad swath of the ways that evolution applies to cancer, and especially how cooperation theory is really important in that. So let's dig in a little bit into some of these questions about how cancer represents a breakdown of multicellular cooperation, and then what that means for how we approach cancer. So if we go back to the origins of multicellularity, we can see that there are five main foundations, really, of effective multicellular behavior. And these include that cells are able to inhibit their proliferation, that they can control cell death, that they can allocate resources effectively. So if you imagine, very early on in the evolution of multicellularity, you have cells that have to start doing things that include transporting resources like to the inside of an aggregation because they're not getting there through just diffusion. So you need to have some mechanisms for getting resources to the cells that need them. Division of labor is another important foundation of multicellularity. We have many different cell types. And then also creating and maintaining an extracellular environment. Now, this is something that oftentimes we don't really think about, but our bodies are not just made of 30 trillion cells. In between those cells, there's a lot of stuff. And there's a lot of stuff that cells are actively making, waste products that cells are actively removing. And there's cells that are sort of moving around in that extracellular environment to maintain it actively. And that extracellular environment and the sort of care for it is another foundation of multicellular cooperation. It's basically, taking care of the environment, taking care of the neighborhood that the cells are living in. And so in this transition from unicellularity to multicellularity, cells evolved to cooperate and coordinate in these ways. And we can think about cancer really as a breakdown in those foundations of multicellular cooperation. Because to have effective multicellular cooperation, you need all of these features. But if we look at the phenotypes of cancer, they're characterized by disinhibiting proliferation, by avoiding cell death, by monopolizing resources, by shirking and co-opting the labor of other cells as opposed to properly doing their jobs and dividing labor. And they also destroy the environments that they're in. They literally will produce acids that break down the extracellular matrix, and that is not only just sort of cellular laziness, it actually can make invasion and metastasis more likely. So it's really a critical issue that really breaks down the structure of the multicellular body in a way that can make the cancer cells more viable. Oftentimes when I kind of get to this point in the talk, I'll get a question from the audience sort of asking, well, how are you defining cheating? How can we say that cells are cheating? Doesn't cheating imply intention? And we know that cells don't have intention. Well, of course, all of this depends on how we're defining our terms. So I'm going to define cheating here as the breaking of shared rules that lead to a fitness advantage for the individual that is breaking those rules. So in the case of human systems, those rules might be things like laws or social cultural norms that people share. People might cheat in those in order to gain some personal advantage. In the case of cancer, cheating is breaking of shared rules, but those shared rules are really instantiated in the genomes of all of the cells in the multicellular body. And they're shared rules because they're there in every cell, and they regulate the cooperation and the coordination of all of the cells in the body. So when cancer cells don't sort of behave according to those rules, then that is the same sort of structural kind of issue, problem that you can have in human societies. And so what happens practically is that you can have mutations that will give rise to these cellular cheaters because they're breaking down these foundations of multicellular cooperation and cheating by signaling for more resources, proliferating, not dying, not doing their jobs, exploiting the extracellular environment. All of those things are governed by genes that can mutate. And then when that happens, those cells can gain an advantage over other cells. And what then happens is that those cells can grow in frequency, and that is ultimately what leads to the initiation of cancer and the acceleration of cancer if it has already been initiated. So let's now take a look at this with a slightly different lens. So we've already talked about how we have these foundations of multicellularity and how we have a breakdown in these foundations of multicellularity when it comes to cancer. And now we can say, all right, well, how does this map on to what we know already in cancer biology about the phenotypes of cancer? What are the characteristics of cancer that clinicians see, that cellular biologists see? What are the features of cancer that have sort of already been established? And there's a framework called the Hallmarks of Cancer that was put together by Hanahan and Weinberg. And that basically describes, these are the characteristics of cancer that we have, or that, scientists, that researchers have already identified. So what we did was we took the Hallmarks of Cancer and we wanted to see how they would map on to these breakdowns in multicellular cooperation, the breakdowns in the foundations of multicellular cooperation. So what you're seeing now on the outside here is the hallmarks of cancer. And you can see that many of them map on to the breakdowns of multicellular cooperation. In fact, they all map on somewhere. The uncontrolled proliferation is that there are 3 hallmarks that map on to that. There are 2 that map on to inappropriate cell survival, 2 that map on to resource monopolization, and then the activation of invasion is something that occurs partially as a result of environmental degradation that can happen in cancer. Although it's not 100% of a map on, it's certainly the clinically relevant issue that can occur because of the breakdown of that extracellular environment. And then we have on the bottom dysregulated differentiation here with 2 stars because this isn't and wasn't a hallmark of cancer when we began this process, but as a result of mapping the hallmarks of cancer onto the breakdowns of multicellular cooperation, we proposed that this actually was a missing hallmark of cancer. And we've now actually been communicating with the original authors of the Hallmarks of Cancer, and we've proposed this, and we're in the process of we have a paper under review looking at this, and also it's a really interesting example of how you can take an evolutionary approach to these kinds of questions, and then turn it back onto cancer and gain some insights potentially into what are the phenotypes of cancer that come from this cooperation theory and evolutionary perspective. So cancer is fundamentally a part of multicellular life, and we're going to dig a little bit into some of the places where we see cancer that are surprising in a minute. But first, I want to share with you a music video that is based on a song that I co-produced with Baba Brinkman. We began this project many years ago. And essentially, for those of you who don't know who Baba Brinkman is, he is a peer-reviewed rap artist. And he's written and performed many raps about evolutionary biology in general, making ideas of evolution really fun and accessible for broad audiences. And we had this idea to create a rap song from the perspective of a cancer cell who was not happy with being stuck in the multicellular body and not being able to be an independent entity. And so this was a really fun project, and the video is not so long, so I'm gonna show it to you now, and then we will loop back around to looking at cancer across the tree of life. So I'm going to share this with you now.
Forefathers were free, but I was born a slave. I keep the memory of freedom in my DNA. I'm talking 700 million years ancient. My ancestor was a eukaryotic free agent, feeding and reproducing and feeding and reproducing and feeding and reproducing and never needing to do things until someone made a cynical wager and traded their independence for division of labor. Damn, I wish I knew who the sellout was. I'm ashamed to be descended from his sellout blood. Multicellularity ain't nothing but a scam. Stuck in one place your whole life, working for the man like you're a liver cell. You stay in the pancreas, hey, congratulations, stem cell. Fascist, the body is a one-party dictatorship, I can't escape it. But goddamn, I can make it sick and spread the dream of freedom like a rumor. Spread it like a Tasmanian facial tumor. So what if I'm a cell from the somatic line? You can stick your limitations where the sun don't shine. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic. The revenge of the somatic line, let them have it. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic. The revenge of the somatic. I used to be a slave myself. I felt senescence hastening before my carcinogenic awakening. A couple 100,000 puffs of tobacco smoke and I just wasn't as open to apoptosis. That's a bad prognosis. I was hit with every tumor suppressant mechanism in the human immune system, but I mutated with it. I was super persistent. Every daughter cell was suitably different. Therapeutic resistance came to me like a beautiful vision. I really thought I was doomed, but evolution assisted with the chemotherapy. So clever and devious, but I was already genetically heterogeneous. You just deaded the weakest. Now competitive release is inevitable, so witness as my fitness increases. Entering untapped niches, I gather the benefits. I'm relentless. I get fed from angiogenesis, ducking T-cell predators. I keep it anonymous so I can exploit the body's weakness and tolerance. I'm a smooth criminal, so I never get caught. I just pimp the system like credit card fraud. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic. The revenge of the somatic line, let them have it. Cause I'm ready to die in the fight to be free. I'd rather multiply than live on my knees. And I'm 2 mutations away from metastatic. The revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic line, let them have it. Revenge of the somatic.
Well, so that is Revenge of the Somatic. And it was so fun, I have to say, working on this project with Baba Brinkman. And I highly recommend, if you get the opportunity to collaborate. Oh, let me make sure you don't keep listening to Baba Brink. You should keep listening to Baba Brinkman, but not during this talk. So be sure to check out Baba Brinkman's work. He has many, many great songs about evolutionary biology and a whole album on evolutionary medicine. So and I have to also say that working with Baba Brinkman on this project, it shifted my thinking to really considering, what does it mean began to look at cancer from the perspective of a cancer cell, or a group of cancer cells. And for those of you who have taken a lot of evolutionary biology classes, or you're, professors and researchers working in evolutionary biology, you know that one approach that can be really useful when trying to understand why organisms are the way they are is to take an adaptationist perspective, which means that you're, considering, well, what are the problems that the organism has to solve in their environment, and what would we then expect, what kind of adaptations would we expect those organisms to have, given that their environments are like this and they have to solve these kinds of problems. And working on this video with Baba Brinkman really put me in that sort of mindset of, okay, well, what is the environment that cancer cells are facing? And what does that mean in terms of what we would expect in terms of the evolution of behavior among cancer cells, not just, sort of thinking of their physiology, but also thinking of their behavior. So let's look at what all of this means for cancer in the tree of life. I've talked to you guys already a little bit about these crested cacti. This is a saguaro cactus, and this is very similar to the first crested cactus that I ever saw. It was also a saguaro, and it really made me realize that there was something that was almost magical kind of about these lovely plants, that they could maybe give us some insights into cancer across life. So it really it was crested cacti that pulled me in to comparative oncology, really. At the working group in Berlin, me and many colleagues sat down every Monday and Wednesday afternoon for hours, talking about what are the big important questions in cancer biology. And for the first 2 months or so, we made lots of lists and had lots of freewheeling conversations. And then we kind of started to prioritize, well, what do we think are the most important things that we should do with our time here? And the consensus was that, this was 10 years ago, actually. And at that time, there wasn't much work at all on comparative oncology. Just, a few small studies had been done at that point. And there was no systematic review of reports of cancer across multicellularity. And so we decided that we wanted to spend a great chunk of our time working on that, which we did, and it resulted in a paper where we looked across all the branches of multicellularity, and we found that in each of these branches of multicellularity, there was cancer reported in the sense that there was invasion or metastasis, and there were also many where there were cancer-like phenomena, even if there wasn't cancer specifically. And so we define cancer-like phenomena as dysregulated differentiation and dysregulated proliferation. So if we dig a little bit into some pieces of this here, in animals, there's many examples of cancer in vertebrates, but also in a variety of other bilateria. And that's the case, for example, in brittle starfish, in clams, in many different mollusks, in insects, and this is it's a very, very common phenomena in animals. In sort of early metazoans, so early animals, Cnidaria, we see loss of differentiation in coral, and we see what seems to be sort of full-on cancer in Hydra, where you do have invasion into what would otherwise be normal tissue. Can look at fungi as well, and we see loss of differentiation and also inappropriate tissue differentiation occurring in fungi. In plants, as we've talked about already, there are lots of examples of fasciation. And these fasciations, they don't necessarily kill the cacti, and they don't necessarily involve invasion or metastasis. But there have been some reports. In particular, there's one of a bacterium-free metastatic growth in a plant. So it appears that cancer-like phenomena are quite common in green algae, which include, of course, many of the things that we sort of think of as plants. But they're perhaps not as common, like full-on cancer might not be quite as common. So if cancer is a problem that is fundamental to being multicellular, then we can ask the question how multicellular bodies have evolved to deal with that, right? So how do they evolve to detect cellular cheating and effectively respond to it? Well, we have a sort of triple layer system here. One of the systems is, our set of systems are cell intrinsic, meaning that they go on inside the cell. The cells will monitor themselves for DNA damage, for proteins that aren't folded properly, for abnormal proliferation or the presence of a lot of viruses. And if those situations are detected, then that changes gene expression in ways that allow the cell to respond, potentially inducing DNA repair, or if things are really bad, actually inducing cell death. There's also neighborhood level cheater detection and response systems. So all the cells in our body are not sort of just independent entities. They're constantly monitoring the cells around them, responding to them. And they monitor a lot of things, including the adhesion among the cells, the broader sort of tissue architecture that they're in, and that extracellular environment that they're in. Is it normal? Are there cells around them that are surviving when they maybe shouldn't? And every cell is taking in signals from neighbors that allow them to monitor those things. And if it seems as though neighbors are not behaving properly, then cells can actually send signals to induce cell death to their neighbors. And if both of those levels fail, we also have system-wide surveillance. And this is sort of akin to, what at least, in humans is, mostly done through immune system surveillance. So what you have is a situation where if there's a region that has abnormal proliferation or like overuse of resources or a lot of waste production, then that sort of attracts the attention of the immune system, and immune cells will sort of evaluate, is there really a problem here? And if so, then, you can get inflammation and a broader sort of response to the problem. So those are the three main ways, three main levels where responses and detection of cheating can occur. And so if those things are working properly, then they limit that cellular cheating and allow multicellular bodies to really embody that cellular cooperation that is, really what makes us what we are. So how is this cheater detection and response actually instantiated? Well, there are many, many genes that are cancer suppression genes. One of them that is particularly interesting is TP53, and I like to call it the cheater detector of the genome. It is really a central node in a very complex cellular information processing network. It's integrating signals from many different pathways, and it essentially decides whether the cell should keep doing what it's doing, whether it's a little messed up and needs to repair its DNA, or whether it is sort of permanently messed up and is a threat to the rest of the multicellular organism. And if so, then it induces it to undergo programmed cell death. And so we can sort of think of TP53 as this decision maker. It's taking all of this sort of, complex information and then, in a sense, trying to decide, like, is this just normal cell behavior? Maybe in a slightly unusual situation, maybe there's some wound healing going on, so there's a little more proliferation than there should be, but ultimately everything's normal. Or is there actually a problem? And that's a complex decision, really, to make. It requires integrating lots of information from lots of different sources, and then enacting changes in gene expression that allow the cell to sort of either get back to a state where it's not a threat to the body, or for the cell to actually commit cellular suicide, which is sort of the ultimate sacrifice in evolutionary terms. So how does TP53 detect cellular cheating? Well, it's tapped into so many of these pathways of cellular gene expression that it's essentially listening in on all of the things that the cell is doing, or many of the things that the cell is doing, and it's able to kind of triangulate, like, is there a problem here, because it's listening to the all of the things that the cell is doing. Now, what's fascinating is that there are organisms that have more copies of TP53 than others. So we humans, we have 2 copies, one from our mother, one from our father. But elephants have 40 copies of TP53, and their cells are also much more sensitive to undergoing apoptosis if they're exposed to radiation, suggesting that they're kind of on a hair trigger, potentially as a result of having more copies of this tumor suppressor gene. In work done by some of our colleagues, they also looked at the evolution of copy number of TP53, and they found that increasing body size was potentially driving selection on TP53. They were able to look at this by comparing the African elephant with woolly mammoth and mastodon genomes to see how the increases in body mass preceded the increases in copy number. And it's not just limited here to looking at TP53 and elephants. If we look at other large organisms, including whales and dolphins, we see that there's positive selection for lots of genes that are involved in monitoring the cell cycle, in cellular signaling and cell proliferation, which suggests that genes for suppressing and regulating cancer are really under important selective pressure in these large and long-lived organisms. And so one way that we can think about what's going on here is that large scale and long-lived multicellularity, big organisms that live for a long time, had to evolve to suppress cellular cheating better than their smaller and shorter-lived cousins. And I like to think of us as our bodies as actually having a sort of collective cellular intelligence that's keeping cancer under control, because we're made of 30 trillion cells. Each of those cells is processing a huge amount of information all the time in order to monitor what's going on inside the cell and around the cell, and in many cases in the rest of the body, in order to regulate our cells to get us to the point where we can survive and reproduce, and hopefully pass along those cancer suppression mechanisms in the process. So I want to take just a few minutes before I open up for questions to talk about what is the future of cancer if we take this sort of evolutionary perspective. And there are a lot of evolutionary informed treatments that can help to control cancer. We can aim to support the cooperation of normal cells, to support cheater detection systems, to try to disrupt cooperation in cancer cells. I won't really have much time to talk about that today, but cancer cells actually re-evolve cooperation with each other in order to better exploit the body. And so this is another potential target for treatments, is to disrupt that cooperation among cancer cells. Can also try to slow down the somatic evolutionary process happening inside the body, because if you even just slow that down, by 50%, that can be sort of enough time that people could live a long and healthy life and die with cancer rather than of it, because they reach the end of their natural life without succumbing to cancer. One other one that I think is extremely important that comes out of taking an evolutionary perspective is to minimize the evolution of resistance. And that's something that Bob Gatenby at Moffitt Cancer Center has worked on quite a bit. And I'll show you a little bit of data from him in a minute. We've also suggested that it can be really important to sort of switch the mindset from trying to kill cancer cells to instead trying to protect the patient and limit the tumor. Because if you approach a tumor just from the perspective of, can we eliminate it at all costs, that often can end up selecting for resistant cells. Now, if it's possible to remove a tumor, to resect it surgically, to take it out, or to treat it with high-dose radiation and have it be eliminated, of course, you should pursue that. But when you get to the point where cancer has progressed and it's not possible to eliminate it, then it becomes extremely important to consider what selective pressures are we putting on the tumor with our treatment. Are we selecting for cells that are resistant? And oftentimes the sort of default treatment of, several rounds of very, very high doses of chemotherapy, that strategy can select for resistance. In fact, let's talk a little bit actually about that here. So adaptive therapy is this approach that Bob Gatenby has put forward in computational models, but also now in clinical trials. And he's found that this controls tumors in ovarian and breast cancer in mouse models. And essentially what adaptive therapy is that you treat the cancer with a dose to reduce the size to a manageable size. And then you only treat it when it grows. So you actually allow the tumor to continue to exist, but you keep it from growing too much. And what that does is that it maintains sensitive cells in the population. So if you look at the bottom panel here, you can see those purple cells sort of hanging around, and that's because the doses are not so high that they eliminate all of the sensitive cells. And because oftentimes there's a cost to being resistant, it requires more energy to, say, pump out the drugs, or there can be other ways that it can be evolutionarily costly to be resistant. Once you then remove the treatment, the sensitive cells have an advantage over the resistant ones, and that allows for long-term control of tumors. This is a slide here with two panels. On the top, you see the individuals who were in a trial where they were using adaptive therapy. And this was on prostate cancer, advanced prostate cancer, that was, it was sort of at the point where other treatments weren't working. So this was sort of a last ditch resort. Would adaptive therapy work? And what you see is that in the individuals that were using adaptive therapy, there was only one who progressed to completely untreatable prostate cancer, while in the bottom panel, almost everybody progressed, and many of them quite quickly. So this is an extremely promising approach, and one that there's currently a lot of efforts to do more clinical trials, but unfortunately, it's hard to fund because there aren't a lot of individuals, there aren't a lot of companies who can benefit from it, because it can be used with any drug, and even any type of treatment. You could use it with radiation, because it's really an algorithm for treatment. And so ironically, the fact that it can't make a lot of money, but is also potentially cheaper, makes it harder to have it go through the process that would, allow us to, as a community of cancer researchers and concerned human beings about cancer, to get this really promising treatment into the clinic more rapidly. Now, what I want to suggest broadly is that the future of cancer really is about living with cancer rather than dying of it. And treatments like adaptive therapy, I think, are extremely promising for that. And, really, this is a reflection also of just the nature of cancer, that, we as multicellular organisms have coexisted with cancer since the beginning. And we're going to continue to coexist with cancer. And if we can leverage our evolutionary understanding of how multicellular life suppresses cancer, keeps it under control, makes it possible for us to, thrive despite the vulnerabilities that we have to it, then I think we will all be able to thrive with the threat of cancer, or even with having tumors that we manage in the long term. And I just want to bring us back at the end here to our cactus garden, and to this quote from Charles Darwin that inspired the name of our garden, Endless Forms Most Beautiful. Darwin said in Origin of Species, "There is grandeur in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." Thank you all so much for your time and attention, and I'll be very pleased to take any questions right now.
Thanks so much, Athena. That was brilliant. Really, really fun, interesting talk, which is not always what you expect from a cancer talk. No, that was really great. And it's also quite nice that you sort of end on quite a positive note, even, just talking about managing tumors and living with them, as opposed to sort of like eradication narratives and, that sort of thing, which really seems a bit sort of futile. I guess one thing that comes to mind, if for that to be a reality, that the early detection is presumably still vital, because there must come a point when you can't live with the tumor or manage the tumor, as you say.
Absolutely. Yeah. So oftentimes very early on, it's possible to resect a tumor, to cut it out surgically, and sometimes you don't even need to do chemotherapy. Sometimes you need to do some, and then you essentially have a cure, because you've caught it before it had time to evolve sufficiently to be able to really exploit the body in a insidious kind of way. But if cancer has progressed, then, you can reach the sort of point of no return where, it's like one way or another, you're going to have cancer cells in your body for the rest of your life. And the question is, what selective pressures are you going to put on them with the treatments that you have? And depending on, the receptivity that the oncologists and clinicians have to really taking an explicitly evolutionary perspective, you might or might not have options available that are taking this explicitly evolutionary perspective. But there's some great resources out there. I believe there's a a website called Uncharted that is like a master class for people who are struggling with cancer and want to learn more about sort of the evolutionary approaches and how to integrate those into their treatment, how to talk to their doctors about them. So I would highly recommend that.
That sounds amazing. So we have a few questions that have come in. Actually, the last question that came in is quite relevant to what we've been talking now. So I'll ask that first, and then I'll go back up to the beginning. This looks a lot like the processes behind antibiotic resistance. Okay, so talking about the chemotherapy, do you think adaptive therapy solutions might work in tandem with research on antibiotic resistance? Should these people be working together?
Yeah, in fact, the community of researchers within evolution and cancer, looking at the evolution of resistance, we're very much in contact with people like Andrew Reed, who work on the evolution of antibiotic resistance, and some similar phenomena really happen across those domains. So, Andrew Reed has shown that there are actually many cases in which using high doses can actually be a very bad thing, even though oftentimes the sort of public health message that's given is like, take all your drugs, otherwise that'll give you the evolution of resistance. But that's not always the case, because it depends on what the selective pressures are, and oftentimes taking high doses and a lot of drugs actually leads to the evolution of resistance rather than the opposite. It's really all about the timing, because as we were talking about, right, like early on, yes, perhaps you can get rid of a tumor with surgical resection, with, aggressive chemo, a high dose chemo, and eliminate it. But if you already have enough diversity and a large enough population, and say a phenotype that is rapidly mutating as well, then you may not be able to get it under control with that high dose, and you could inadvertently make a problem that's harder to solve by approaching it so aggressively.
Thank you. And that reminds me, actually, the NHS in this country has now changed its messaging around antibiotic. They no longer say complete the course. They say something like, listen to your doctor. So that narrative is slowly shifting. It's really hard to change those public health messages, though. It really is.
Yeah.
Okay. Oh, yes. I forgot, if I click on the question, it comes up for you too. So this is a question from earlier on. To what extent are plants good models for human interactions with cancer?
So maybe I don't understand exactly. Are they asking if like looking at plants can help us understand just cancer in humans? Is that the...
I guess so. It's just
someone in
the thing. Yeah. I think it came up earlier on when you had the cacti, I remember.
Yeah. So I think that that plants are, the biology is really different from animals. It's just unbelievably different. And so I wouldn't say they're a good model system, but they're a system where the biology is sufficiently different that you can see different kinds of phenomena, and it makes you think differently about cancer and the constraints on cancer. And plants can be very genetically heterogeneous within. They can accept, grafts. Many of them can just bud off and start, new plants. So their biology is just so different. But they nevertheless are able to sort of manage cellular cheating effectively. And so I think that taking the sort of broad perspective where we examine systems that have biology that's different than humans can help us to understand really well what are the kinds of mechanisms that are similar, what are different, and are there maybe parallels of things that you see in plants, but they're instantiated differently in humans. It's sort of like a little bit of perspective taking there, and I think that plants are a less intimidating way of starting to engage with cancer. And many of the cancer-like phenomena in plants are really just visually intriguing. Some of them are gorgeous. like sculpturally beautiful. I mean, so it's a way to visually engage. You can sort of see, okay, like I can tell these cells are replicating, and they're the growth tip is like different than it is in a normal plant, and okay, and that's maybe what happens in stem cell compartments in the body, but you don't see something that's beautiful if you have a cancer happening inside the human body. I don't think I've ever seen a resected tumor and I was like, oh, that's beautiful, right? But with with plants, many of those fasciations are gorgeous. So I think that what it offers us is not necessarily a direct model system for anything, but a new kind of perspective that can be helpful in other ways.
Thanks. Okay, next question. What causes the cheating to start? How does it all kick off?
Great. That's a great question. So, in order to sort of see where the cheating begins, we have to start with these foundations of multicellular cooperation. So, in order for multicellularity to evolve, right, you had to have mechanisms for making sure that this cooperation was happening, and making sure that if cheating occurred, that it would be suppressed. And so you have a lot of genetic mechanisms that evolve to keep the cooperation happening and to limit that cheating. And so when mutations occur, then that can interrupt the cooperation. It can interrupt the cheater detection. And one other thing to keep in mind is that our bodies are constantly, like cells are constantly dividing because we're replacing our tissues, and so anytime there's a cell division, there's an opportunity for a mutation, and that mutation can then lead to a breakdown of that cellular cooperation, a breakdown of that cheater detection, and then once that's started, if it confers an advantage to the cell that has it, then that cell is going to increase in frequency in the population, and that that's a evolutionary process inside the body, somatic evolution, favoring those cells that are exploiting the body that they're in.
Great, thanks. Interesting to hear that Hydra get cancer too. Given their very long lifespans, how does the cancer not kill them, or does it?
So I don't really know that much about the details of Hydra. I think that it might kill them, though. I don't think that they're able to necessarily survive.
I think they're like the poster species for long lifespans, aren't they? Okay. Is cheater detection and suppression the main function of TP53? What would happen if we introduce more copies to humans as a treatment?
Yeah. Well, so I look at TP53 as this sort of cheater detector and responder. A lot of people call it the sort of the seat of the cell cycle control, right? It's like it determines if the cell is going to continue to divide, if it's going to, slow down its cell cycle, do repair, or if it's going to apoptose. And so when I see that through the lens of, cheater detection and response. But some people sort of think of it as like a cell cycle regulator. And there's actually a lot of efforts to see if it's possible to introduce more copies of TP53 as part of treatment. Joshua Schiffman at Utah has been working on that for a while now. I'm not sure what the current status is of that, but certainly look into Joshua Schiffman's work if it's something that you're interested in.
Wow, great. Okay. I have another question here coming through on the different documents. I can't put it in there. From Grażyna Jazienska. Great talk, Athena. Is your work relevant also to cancer prevention?
Yeah. So, we can think about these same issues of supporting cellular cooperation and, doing the sort of proper cheater detection in the context of preventing cancer or reducing the risk of cancer, right? So if we can support our bodies in making sure that those foundations of multicellularity are occurring properly, that cheating is not taking off, then we can do a better job of preventing cancer. And I think one easy thing that we can all do, or two or three easy things that we can all do, are thinking about our diets, our general sort of exercise patterns, and other lifestyle things like sleeping well, eliminating light at night, things like that. We know that if you are you're eating properly, not only are you if you have lots of fruits and vegetables, not only does that we know that decreases cancer risk, from epidemiological studies, but it also changes a lot of the processes happening in your body in terms of how cells are interacting with each other. In the case of, both eating and exercise, how microbes are interacting with our own cells, and sort of promoting in general, an environment of cellular cooperation in your body, reducing conflict among your cells and between your cells, and microbiome. All of those things are really important for making it just less likely that those cellular cheaters will arise. If you don't have as much inflammation, then you're going to have less potential for mutation. And if those cheaters do arise, it'll be less likely that they'll expand. So there are actually a lot of things that are very easy to do and accessible that can reduce cancer risk. And probably, a lot of the mechanisms for why those work have to do with the fact that they're supporting our bodies in their, cellular cooperation and cheater detection systems.
Wow. Yeah, exactly. The microbiome is also such a fascinating, when you think about the cooperation within the body, right?
Absolutely. Yeah. Yeah. And there's, lots of evidence that there's, certain microbes that are more associated with cancer risk than others. And that, part of why our diets are affecting our susceptibility to cancer is related to the ways that they're changing our microbiomes.
Okay, question from Gabriella. Do you think that the language we use around cancer, fight, battle, influences public opinion and maybe directs research to fight cancer rather than understand it and treat it with an ultimate perspective?
Absolutely. Gabriella, that's an excellent point. I think that when people are sort of in this mode of, fighting and seeing cancer as like a war, as a battle, then the only option seems like just winning, right? Which usually means just eliminating the cancer. And that can, I think, lead people to a mindset where it's very hard to even consider the possibility of coexistence, of actually tolerating a small controllable tumor for a long time and having a decent quality of life with that, rather than trying to eliminate it so then you can be cancer free, except not really, right? If the cancer's already at the point where it's sufficiently genetically heterogeneous and the population size is large enough, and all the parameters of evolution are not working in your favor as the individual who has cancer. So I do think that it's important to change the language that we use around cancer. And I just like to use the phrase of like living with cancer. I think it applies both to how we think about maybe a patient who has cancer that's sufficiently advanced that it's going to be a part of the rest of their lives. But I think it applies to the societal level, to our cultural level, for us to be able to, as a society, live with cancer, to acknowledge that it is something that is present, that's a part of many people's lives, and that we also sort of societally have to adapt to that, rather than sort of seeing anybody with cancer as a like a soldier in the war on cancer. That's not helpful for most people who are struggling with cancer.
That's right. You've got this kind of narrative and this really, fighting the good fight and all that. And it's still kind of taboo in a lot of ways as well.
Absolutely.
Right. Question from OP. Do you think there are similarities between viruses and cancer cells and aims?
So I think maybe the question is like, what's the, do viruses and cancer cells have some similarities?
Right.
And yeah, and absolutely. So there are actually quite a few cancers that are initiated by viruses because the viruses will sort of induce, changes in cells that lead them to have a cancerous phenotype. In general, whenever you have a sort of an entity that is able to hijack another entity, right? So like viruses, cancer cells, and agents of infection in general, if they're able to, sort of divert resources from the organism, from the sort of aims of the organism, right, to survive and reproduce and thrive, to instead promote the evolutionary interests of that sort of smaller entity that's doing the hijacking, then they're able to gain an evolutionary advantage, and then of course that evolves. Because it's effective at exploiting the host. So both viruses and cancer cells are cheaters of a certain kind, right? That are hijacking multicellular bodies and other entities for their own aims in quotes, because obviously they're not consciously feeling like they're trying to accomplish these things, but evolution makes a lot of organisms look like they're trying to accomplish things, so.
Yeah. Well, thank you so much. I think that's all the questions now. Yeah, that's been great. That's been so interesting. Really good to have you here. Really here to help us celebrate Darwin Day Week. Yeah, thanks very much. I'm going to end the broadcast now.